Hello dear students, I welcome you all in this digital classroom. Today in this video we are going to discuss about the enzyme. We will see what are the different nature of enzyme and different factors that affect enzyme activity. We will discuss and we will see why these enzymes are able to increase the rate of reaction, or how the enzymes are able to convert the substrate into a product at such a high rate. Okay, so conversion of the substance into a product, or different substrate into a product, increases into manifold, means the rate of reaction becomes very high in presence of enzyme. So we will see how it is possible, what is the nature of enzyme activity, and what are different factors that affect the enzyme activity. Let's begin. To understand the enzyme action, let us understand what is enzyme. So enzymes are the proteins that that helps in the catalytic activity, or that helps in catalyzation of a chemical reaction. In fact, in a biological system, all the chemical reactions are catalyzed by enzymes, right? Since enzymes are protein, they have different structures, including the primary, secondary, as well as tertiary and quaternary structure. So when they have these different types of secondary, or the quaternary, or the tertiary structure, there are different crevices or pockets are formed. Okay, there are grooves that are formed, which are called the active site. So these active site are the site for the binding of substrate on the enzyme, right? So the function or the action of enzymes are very specific to their substrate. There is a specific enzyme for every substrate, right? It's like a lock and key method, or it's like a lock and key, that every lock has its own specific key. Similarly, every substrate has its own enzyme, or the enzyme is specific to the substrate due to the type of crevices, or the type of pattern of the crevices that they have. So it should exactly fit into that crevices or the grooves onto the enzyme. Okay, so this is how the very first step that happens, or that occurs, is the binding of enzyme with the substrate. Okay, so in this way the enzymes are able to function actively on the substrate, that is to bind on it due to the crevices or the pockets that are present on the enzyme. So the binding of this substrate and the enzyme, it forms a complex which is called enzyme substrate complex, right? So the enzyme substrate complex, after it is formed, now it starts to undergo the chemical reaction, right? In a chemical reaction, the substrate that undergo the reaction gets converted into a product, and during the process the substrate completely loses its chemical property and gets converted into a new substance, which is the product, right? So in this way the enzyme substrate complex undergo the reaction and forms a product, and once the product is formed, the enzyme is released, right? So you can say that in a reaction, enzyme is not consumed, rather it is used as a helper, you can say, or it is used to enhance the rate of reaction. Even though the chemical reaction can happen without enzyme also, without the presence of enzyme, the activity or the reaction can happen, but it is very slow as compared to the presence of enzyme. So the presence of enzyme increases the rate of reaction by making an enzyme's substrate complex, right? So this complex will enable the reaction to become faster in the formation of the product, right? So in this way the enzyme help in a chemical reaction. So we will see now in a graph how this enzyme activity, or how does the enzyme work by forming the substrate, by forming the enzyme substrate complex. As you can see here in this chemical reaction, this E stands for the enzyme and S stands for substrate. So there is an equilibrium, means the enzyme and substrate, they bind to form an enzyme substrate complex. Okay, then this substrate get transformed into product, that is enzyme and product, and later on the enzyme is free from the product. I mean the product is separated from the enzyme. Now this enzyme can be ready for the next reaction to happen. So here we can understand that in a reaction the enzyme is not consumed, it is only used to enhance the rate of reaction. Now the same thing let us understand with the help of this graph. In this graphical representation, this is the to understand the concept of activation energy. Okay, so here in the y-axis, this is the potential energy, the energy that is required by the enzyme to undergo the binding. Okay, and here in the y-axis, this is the progress of the reaction. So during the state when the substrate is bound to enzyme called the active site, right? When the substrate binds with enzyme in its active site, a new structure of the substrate called the transition state structure is formed. So very soon after the expected bond breaking or making is completed, because in a chemical reaction either the bond formation will be there or there will be the breaking of the bond. So after the expected breaking or making of the bond is completed, the product is released from the active site, means the product will be released from the enzyme. In other words, we can say that the structure of substrate get transformed into a structure of product. This is what we have seen in the reaction also, like how the product is separated from the enzyme. And during this entire process, the pathway of this transformation must go through a transition state. So in this graph here you can see the transition state. Okay, so this line shows that from here, after providing a certain potential energy, suppose if this starts from 0, at certain point after the energy is being provided, it will start to bind with the enzyme. I mean the substrate will start to bind with the enzyme. Okay, so it has to reach a transition state. So after this transition state, it will no longer form the complex, but it will start to release the product. Okay, in this way the product will be released, right? So here you can see that the stability is related to the energy status of the molecule or the structure. Hence when we look at this picture or this graph, it looks like something as figure 9.6 here in this, when the y-axis represent the potential energy and the x-axis represent the progress of the reaction of the transformation of the substrate into product. Here the energy level difference between substrate and the product is different, right? Here you can see that the energy level difference is seen in substrate and the product. If product is at lower level, then S. If product, when the energy level is less in case of the product, right, the reaction is an exothermic reaction. Exothermic reaction means when energy is released, right? One need to supply energy by heating in order to form the product. However, whether it is an exothermic or spontaneous reaction or an endothermic or energy requiring reaction, the S has to go much higher energy state or transition state. The difference in average energy content of S from that of this transition state is called activation energy. So it requires an activation energy to go the transformation. So enzyme eventually bring down this energy barrier and making the transition of S to P more easily, right? Here these two curve shows in presence or in absence of energy enzyme. You can see this curve, activation energy with enzyme and activation energy without enzyme. So activation energy required is much more when the reaction is without an enzyme. So activation energy is much required, whereas here in low activation energy comparatively the substrate can be transformed into product, whether it's an exothermic or an endothermic reaction. So this graph or this curve, this line also tells us about the action or the activity of enzyme, right? So in this way we can understand here about the activation energy with enzyme and activation energy without enzyme, and this is the transition state. The transition state at which the enzyme binds with the substrate in order to transform into product. So it must undergo this transition state to be transformed into a product. Okay, so this is about the enzyme action. Now here we will see the catalytic cycle of an enzyme action can be described in the following step. Here are the steps that explain about the catalytic cycle, means how does the transformation occurs or how does the reaction happens. First, here the substrate binds to the active site of the enzyme fitting into the active site. As it is already discussed that at the very beginning the active site in the enzyme binds with the substrate. I mean the substrate binds onto the enzyme in its active site. Then secondly, the substrate including the enzyme to alter its shape. The binding of the substrate induces the enzyme to alter its shape. The shape of the enzyme gets changed due to the binding of the substrate, fitting more tightly around the substrate, right? Then third is active site of enzyme, now in close proximity of the substrate, breaks the chemical bonds of the substrate and the new enzyme, that is product complex, is formed. Means enzyme plus product is formed because of the breaking of the chemical bond, or maybe making of the chemical bond. Because of the chemical activity, a new complex will be formed, that is called enzyme product complex. First, during binding it will form enzyme substrate complex. After binding it completely, it will start to undergo change, okay, maybe breaking or making. Then after the change it will form the other complex called the product complex. Then lastly, the enzyme releases the products of the reaction and the free enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. So in this way the catalytic cycle is continued, right? So enzyme remain unused during the entire chemical reaction, right? So this is about enzyme action, means how does the enzyme work on a substrate. Now let us see what are the different factors that affect enzyme activity. The very first factor, there are different factors that may affect the enzyme activity, that may affect the binding as well as binding and also the transformation of the substrate into product. So the first factor that may affect is the temperature and the pH. So enzyme generally they function at a very narrow range of temperature and pH. Generally at high temperature the enzyme gets denatured and become non-functional. So enzyme show its highest activity at a particular temperature and pH called its optimum temperature and optimum pH. Neither very low nor very high. It has to reach that optimum temperature. So activity declines both below and both above the optimum value. It should not be very high or be very low. So at a very optimum temperature. Low temperature low temperature preserves the enzyme in a temporarily inactive state, whereas high temperature destroys enzymatic activity because proteins are denatured by heat. Right, so in this way temperature and pH can affect the enzyme activity. Here in this graph you can see this is number A. Number A is the activity of pH, number B is temperature. So enzyme activity is high at this level. It's the highest at this level when pH is at optimum point, okay, and it goes down gradually. Likewise in case of temperature as well, it goes high at its optimum level and then goes down gradually if the temperature goes down, right? So in this way temperature and pH can affect the enzyme activity. The next factor that may affect the enzyme activity is the substrate. The amount of substrate present may determine the binding of the enzyme. See here in this graph, the amount of substrate determines the formation of enzyme substrate complex. The more the amount of substrate, the rate of reaction goes increasing. Means the velocity of reaction increases with the presence of substrate up to certain point, which is called Vmax, velocity at maximum. Velocity means the rate of chemical reaction is at maximum, right? So it depends upon the availability of the enzyme molecule and the substrate. Since the enzyme molecules are fewer as compared to the substrate, it will bind, the enzyme will bind completely until the last enzyme molecule that is present in the reaction. Then it will start to form the product and it will stop to bind with the substrate, because there is no longer the presence of enzyme, since the enzyme availability is less as compared to the substrate. So at this point, now the enzyme substrate complex formation will be stopped at Vmax, right? And it will start to get converted into enzyme and product. And once product is free from the enzyme, now enzyme can be made available for the next cycle of reaction and to bind on the substrate. So in this way, the substrate availability also can affect to the enzyme activity. Okay. Other than temperature, pH and substrate concentration, there are some other substances or presence of some specific chemicals also may affect the enzyme activity, or also may affect the binding of the enzyme with the substrate. So when binding of the chemical stuff of enzyme activity, the process is called inhibition. Okay, the chemical reaction is shuts off. The binding or the presence of certain chemical with the enzyme may shut off the reaction, which is called inhibition, and the chemical is called inhibitor. Okay, so please remember, the process is called inhibition, that shuts off the reaction, and the chemical is called inhibitor. These inhibitors are closely similar in their structure with the substrate and inhibits the activity of enzyme. Means there will be a competition, because if the structure of the inhibitor is similar to that of substrate, then definitely the substrate the enzyme will tend to bind with the inhibitor as well. So such is known as competitive inhibitor. Okay, so inhibitor can be of similar structure to the substrate, which will degrade or which will decrease the rate of formation of enzyme substrate complex due to its close structural similarity with the substrate. So in this case, the inhibitor competes with the substrate for the substrate binding site of the enzyme. Consequently, the substrate cannot bind, and as a result, the enzyme action declines. So example, inhibition of succinic dehydrogenase by malonate, which closely resemble the substrate succinate in structure. This is an example of competitive inhibitor. So such competitive inhibitors are often used in the control of bacterial pathogens. So this has some applications as well in controlling the pathogenic activity onto the organism. So here we have come to an end of our discussion. So we have learned in this video about the enzyme action and enzyme activity, how different factors can affect the enzyme activity, including the temperature, the pH, the concentration of substrate, and also some specific chemicals which are called the inhibitors that may resemble the structure of the substrate and may affect the enzyme activity. So in this way we have learned how enzyme helps in increasing the rate of reaction by forming an enzyme substrate complex and then an enzyme product complex, and later the enzyme is made free for the next cycle of reaction. So with this I end here. I hope that this video will help you in conceptualizing the topic as well as in learning the things well. So thank you very much and hope to see you in my next video. Till then, bye bye.